So, good evening and welcome to Basic Science Lights the Way. I'm Steve Kahn. I'm the Dean of Mathematical Physical Sciences here at Berkeley, and I want to thank you personally for choosing to spend your evening with us. Tonight, we will be joined by an astrochemist, a biologist, and an earth scientist who are all conducting research in the emerging field of geobiology here at Berkeley. In a broad sense, geobiology is the study of the coevolution of the Earth and of its constituent life. It employs both the information preserved in and by the biosphere today and recorded in the rock record. The interaction of life with the Earth works in two directions. Life has had an enormous impact on the chemical and physical evolution of the planet, and in turn, global changes in surface environments have left their imprint on the genomes of every living organism. Embedded in life's biomolecules are clues to Earth's history spanning back billions of years. They yield information on the Earth's evolution and tell us how we got here. Every major climatic and tectonic event on this planet can ultimately leave its imprint somewhere in the genome. And in turn, new innovations in genomes can profoundly impact the Earth. Our speakers tonight are working across the Earth and biological sciences to explain how the Earth and its biosphere have co-evolved. Now, to kick things off, let me introduce our moderator this evening, Professor Dipti Nayak. Dipti is an assistant professor in the Division of Genetics, Genomics, Evolution, and Development in the Department of Molecular and Cell Biology. Her research group uses genome editing to study archaea, which are single-celled microorganisms and also some of the oldest life forms on our planet. Her group specifically focuses on archaea that produce and consume methane, which is one of the greenhouse gases causing our planet to warm. Thank you for being here this evening as our moderator, Dipti, and let me turn it over to you.
Thank you for the introduction, Dean Kahn. I'm happy to be here with you all this evening. I've participated in quite a few of these events at this point, and they're always fun and informative. As Dean Kahn mentioned, I study microbes that belong to a domain of life called the archaea. And some of these archaea produce methane gas as a byproduct of their metabolism. And for that very reason, they're called methanogens. These methanogens have consistently been some of the most important members of the biosphere for the entire history of life on Earth. From likely playing a very central role in the emergence of life billions of years ago, to contributing to some of the most pressing issues today that are facing modern human society. In the context of the topic today, geobiology, what is especially interesting about archaea and the methanogens that my lab studies is that they are the progenitors for humans and all the macroscopic life that we see around us, which includes insects, other animals, plants, fungi, so on and so forth. So when we learn more about these organisms, these methanogens, we also learn a little bit more about ourselves, our planet, and also how life on Earth evolved over a billion years ago. So that's what I do, but I want to shift gears a little bit and I want I want to introduce you to our first speaker, who's going to tell you more about what conditions may have supported life elsewhere on our planet, on our universe. And so our first speaker this evening is Jenny Bergner, who is an assistant professor of chemistry and astronomy. Jenny is interested in the chemistry accompanying star and planet formation and how this shapes the compositions, properties, and potential habitability of newly formed planetary systems. She uses telescope facilities to explore the molecular composition of planetary building blocks around other stars, and she also uses lab experiments to characterize the chemical and microphysical behavior of molecules under interstellar conditions. Jenny, thank you so much for joining us this evening. The floor is yours.
So I'm excited to be here and set the stage for what our understanding of the initial composition of Earth was, as well as other planets and rocky planets in other solar systems, and how this may influence subsequent biology that can evolve on these planets. So I want to start with this very beautiful image of our own Earth from the recent Artemis mission. And we picture our own Earth and we think of it as this very lush, water-rich planet with all of the ingredients that we know led to the emergence of life. And when we think about the chemical building blocks that life itself requires, given the example of our own Earth, we think of things like water, carbon, nitrogen, phosphorus, these molecules and elements that show up in all life forms here on Earth. So I'm going to use the term volatile to describe these kind of life-important molecules and elements. This is to distinguish it from refractory materials like silicates and iron that make up the kind of core of and mantle of our Earth, but the volatiles are the parts that are very reactive, very participatory in biology and biochemistry. Okay, so we think of these volatiles as critical for life, but actually, from a kind of mass budget perspective, the Earth turns out to be a very volatile, poor planet. So this is an image if you took all of the water on Earth and kind of collected it into a drop and compared it to the rest of Earth, it would be just this small kind of bubble in comparison. And so, in fact, Earth is really a dry, rocky, silicate ball with a very thin veneer of liquid water on the surface. The same is true for carbon. So this is a plot showing the amount of carbon in Earth compared to other bodies in the solar system. And you can see compared to the sun and the building blocks of our solar system that Earth is depleted in carbon by 99 up to maybe 99.99% with respect to the building blocks. So although volatiles are critical for life as we know it, we need a kind of intermediate amount of volatiles for Earth itself. And we think that there actually could be planets with too much volatile material for life to be easily supported, at least in the way that we think of it today. So an example is something like an ocean planet where you have so much water that is delivered to the planet that you have oceans that are miles deep. There's no land to speak of, and I believe we'll hear in the second talk about the importance of land and shorelines and the emergence of life. Another example is something like a soot planet where there's so much carbon inherited by the planet that its atmosphere is dominated by these haze and smog particles that would make it, again, a lot less hospitable than our own Earth. So it's interesting to think that Earth may have inherited a sort of sweet spot in terms of its volatile composition. We had enough things like water and carbon to kick off life and sustain life, but not so much to turn into something like one of these ocean or soot planets. And so within the context of thinking about geobiology, what I and my research bring to this question is thinking about what actually determined the initial abiotic composition of the Earth. How did these initial building blocks set the stage for subsequent prebiotic and biotic chemistry? And how do we generalize this to rocky planets around other stars? So our approach is to study the process of planet formation to understand the answers to these questions. So we focus on a region of space that we call protoplanetary disks, which are the sites of planet formation around other stars. So they're disks of the gas and dusty material that coalesces to form planets around newly formed stars. So we can look at these protoplanetary disks and get an inventory of the compositions of the building blocks that will eventually form mature planets in these other systems. Okay, so we use a couple of different approaches. One is observational, so we can use really powerful telescopes to inventory and map the volatile, icy, and gaseous material that is available in protoplanetary disks. And then the other approach that we use is experimental. So we can characterize the behavior of volatile molecules under the very extreme physical conditions that we expect to operate in these planet-forming environments. And I'll give a little example of both of these approaches. Okay, so first I'm going to start by talking a little bit about how we can use observations to answer these questions. So James Webb is a new telescope facility a couple years old now. And it is allowing us to look at the icy component of protoplanetary disks for the first time. So we have this kind of cartoon picture of a protoplanetary disk on the left. This is an actual image of one of these protoplanetary disks viewed with James Webb. So this is a solar system that is in the process of forming that we can actually see and spatially resolve. So one of the really powerful things about James Webb is it gives us access to the ice. So this is kind of a more appropriate picture or cartoon of a protoplanetary disk. Throughout most of the disk, the volatiles are present in this frozen icy form because the temperatures are so low. And prior to James Webb, we had no way of characterizing the icy composition of disks. So we can do this with spectroscopy. A spectrum like this, we're looking at the amount of light that the telescope receives as a function of wavelength. And we can see that there are characteristic absorptions or sort of attenuation of the flux at positions that tell us about the molecular composition of this material. So for instance, at 3 micron, this is where water absorbs. So we can tell that there is water ice present based on this feature. At around 4.5 microns, we have CO2, and then carbon monoxide has a nice deep absorption as well. We can also detect rare isotopologues. So this is 13 CO2, and we'll hear from Daniel about the use of these rare isotopes to understand geobiological processes here on Earth. So some of the questions that we're excited to be able to answer with this facility is actually constraining the abundance and composition of icy planetary building blocks. So without going into too much detail, we can basically look at the shapes of these absorption features in the spectra. We can compare them with lab templates, and we can learn kind of compositional information. So a more cartoon image that we might develop is thinking about the different components of ices that are present and how the molecules are interacting with one another within protoplanetary disks and how that affects their distribution and chemistry. We can also visualize how the ices are distributed. So I showed you this image before. That's the distribution of the small dust particles within the disk, but we can also make maps of where the ice itself is. And so here the color map is showing you how ice-rich these different lines of sight through the protoplanetary disk are, and this helps us to understand what types of planets might inherit different ice reservoirs. Okay, so we can also use lab experiments to characterize analogs of these interstellar icy dust grains. So this is the experiment that we've built here at UC Berkeley. We operate in ultra-high vacuum conditions, so we're 13 orders of magnitude below atmospheric pressure, and we're operating at cryogenic temperatures, so just 10 degrees above absolute zero. And if you zoom in on this viewport, you can see this is our cold sample holder. There's a little smudge in the center. That's actually an interstellar ice analog. So analog material to the icy volatiles that are present in these planet-forming environments. So one of the questions that we're really interested in from these lab experiments is characterizing the budget of volatiles present at different distances from the star. So in our own solar system, we know there's a big difference between planets that formed close to the star, the rocky planets, versus planets that formed further out in the solar system, the giant planets. And that transition is set by the position where water freezes onto the surface of dust grains. So the water ice line, we call it, interior to it, you have rocky planets, exterior to it, you have giant planets. But you could imagine that for every molecule, it will have its own ice line, and the positions of these ice lines will determine the detailed composition of the planets that form. So these positions of these ice lines are sensitive to the structure of the ice. This is a cartoon showing a complex ice grain structure where you have a silicate core with different ice components, and then the volatiles are sublimating at different points. So we really want to understand the structure and these microphysical processes that take place within this icy material. that can determine the exact position of these ice lines, which in turn will potentially dictate whether you form something like a Jupiter versus something like an Earth, and has obvious consequences for habitability and the inventory of volatiles on a planet. So this is something we can make measurements of. This is just an example showing a complex ice mixture with many different components, and we can see at what temperature different components sublimate and use that to understand and sort of map back what the budget of volatiles is at different positions in the disk. Okay, so that's kind of wrapping up. I just wanted to show this picture again and think about how we can use what we know about the process of planet formation and the chemistry going on during the formation of planets to inform our understanding of the composition of Earth and the building blocks that are available for prebiotic and biotic chemistry. So with that, I'm happy to take any questions.
Thank you, Jenny. It looks like, let me start with one question. Do you think life is possible on other types of planets, say the ocean planets that you mentioned at the start?
Yeah, it's an interesting question. The life would have to be very different from what we have here on Earth, obviously, in order to evolve. These oceans have extremely high pressures. The greenhouse warming effect on these water worlds is probably very severe. So I don't think we can rule out that life would exist on something like an ocean world, but it would have to look very different than what we have here on Earth.
Okay. And then kind of following up on that, what are the prospects for life forms that aren't either water-based or carbon-based?
Yeah, this is a fun question. So again, I think I'm not a prebiotic chemist, so I won't say that it's impossible, but I will say that there's a lot of water and carbon and these building blocks that we need for life as we know it on Earth. And from a kind of cosmic abundance standpoint, you just have a lot of material to work with. So I'm kind of the opinion that, like, why would something use like silica or silicon if there's so much water and carbon around?
Yeah. Yeah, I remember an X-Files episode on that. Uh, and then one other question that's coming from an audience member specifically talks about the ice line that you showed. So the question is, does the ice line CO2 line distance depend on the mass of the stars? So for other stars beyond the sun, would that line show up differently?
Yeah, absolutely. The more massive the star, the more luminous it is, and that will push the position of these ice lines further out in the disk.
Okay. And then maybe one last question before we move on. So you mentioned the 13C isotope, the rare isotope of carbon dioxide. What do you learn from detecting these rare isotopes? And this is also kind of like leading up to one of our next talks.
Yeah, so we'll hear a lot more about this from Daniel, but the isotopes in a kind of astronomical context tell us a lot about the physical processes of where the material came from, at what point and under what conditions the molecules formed. So we can learn a lot about the origin and history of the molecules that we see in protoplanetary disks by characterizing their isotope ratios. And then projecting forward, we can also learn about the kind of initial budget of these isotopes on a planet like Earth that then sets the sort of T equals 0 for someone like Daniel to look at perturbations to those ratios.
Awesome. Thank you so much, Jenny. So we'll have some more time for questions at the end. So to all the audience members out there, please remember to add your questions to the chat. And so with that, we'll turn to our next speaker, Seth Finnegan, Professor of Integrative Biology. Seth's work revolves around understanding the relationship between environmental change and biodiversity change in our global oceans. And he looks at the origin of animals more than 500 million years ago, all the way from there to the anthropogenic impacts of the recent past. This includes understanding the causes and the consequences of mass extinction events, especially the first major mass extinction event following the evolution of animals, the late Ordovician mass extinction. And we'll hear a lot about that today. So Seth, can you tell us about your research?
Okay, great. Can everybody hear me okay?
Yeah.
Okay. So I'm a paleontologist, so I study fossils, and probably a lot of you, when you hear the word fossil, you think of something like the picture on the left here. So this is, some of you probably know this specific fossil. This is a Tyrannosaurus Rex that is mounted in the lobby of the building I'm in right now, the Valley Life Sciences building here on campus. Um, and I think dinosaurs are just as cool as anyone. But I'm going to try and convince you that the fossils on the right here, these humble clams and snails and other marine invertebrates like them, are a fossil record just as worthy of study, maybe even in some ways more worthy of study. And there's a lot of reasons for that. And that begins with, and I say particularly, I'm interested in marine invertebrates. So every lineage except vertebrates that lives in the oceans. And there's many reasons for that. And one of them, as Jenny's talk really nicely set up, sort of the existence of liquid water on Earth was probably a necessary boundary condition for evolution of life, certainly as we know it. We think that life originated in the oceans. And in fact, a lot of the diversity of life is still confined to the oceans. So many evolutionary lineages that have never managed to make it from the oceans to land. The oceans also play a critical role in modulating Earth's climate. And so the history of the coevolution of Earth's climate and life is very much a marine story. There's also a sort of more prosaic reason for that, which is that marine invertebrate fossils are very abundant in the geological record. So if I want to learn more about something like that T-Rex, I have to go to Montana and spend, a long time searching. And if I'm lucky enough to find one, excavating it and bringing it back and studying it is a very expensive multi-year project for one specimen. Whereas if I want to learn more about marine invertebrates, I can find them 15 minutes from campus, or here an hour and a half from campus on the coast near Santa Cruz. So this is a place where we take students, these are some Berkeley students right here, to look at the very abundant fossils, in this case, that are about 6 million years old, of what our local marine ecosystems were like. And in fact, the largest biologically built structures on Earth, arguably, are really sort of nothing but big piles of marine invertebrate fossils. So this is the island of Moorea near Tahiti, and some of you may know that Berkeley has a field station here. It's in the field of view on the right-hand side of this bay. And I'm very privileged every other year to teach an intensive field research course there. And one of the things that we ask the students to do soon after we get there is snorkel over and grab a handful of sand from these beautiful reefs that you can see in the foreground here, and bring that back, and they throw it under a microscope. And you can see that this sand is just nothing but trillions and trillions of fragments of the skeletons of organisms, of animals and algae that create that reef and inhabit that reef. And so this is literally, sort of a record of a whole ecosystem in a handful of sand. And eventually, if these reef sediments are buried deeply enough, they will turn into a rock called limestone and enter the long-term geological record. And so there are whole enormous areas, whole mountain ranges on Earth that are made out of limestones that formed in this way. So this is another place that we've taken Berkeley students. These are the Guadalupe Mountains, Guadalupe National, they're in El Capitan National, no, El Capitan National Park in West Texas. And what you see here is an exposed cross-section through a reef. So this records an enormous reef system that grew about 260 million years ago. And again, if the students walk up and put a hand lens on these sediments here, they can see that they are made up out of the skeletons of the very different organisms that were inhabiting and building reefs 260 million years ago. So there's a record of it that these reef ecosystems have existed for a very long time, but the evolutionary players have changed through time. So one reason is just that these fossils are very abundant. Another reason is that they're incredibly information rich. So from studying them, we get the kinds of information that you probably associate already with paleontology, information about the evolutionary history, the ecology, how they grow, how they function, rates of speciation and extinction through time. But the chemistry of these fossils, which are grown, precipitated from seawater, actually are there's an incredibly important archive of environmental conditions through time. So this is actually the primary way we know about things like Earth's climate, temperature, pH, even chemical weathering rates and tectonics, all come largely from actually studying the chemistry of fossils. And one of the reasons I was really excited to come to Berkeley is that Berkeley has been a real leader in developing these kinds of methods. And you're going to hear in the next talk from Daniel Stolper, one of the current leaders in developing geochemical methods for making inferences about the environmental and biological history of Earth. So we can put these two complementary types of information that come from the same fossils together to understand how Earth and life have co-evolved through time, which again is sort of the essence of geobiology. So this here is sort of showing the taxonomic diversity of different major groups of animals through time. And I've superimposed that on our current understanding of global climate through time, which again comes largely from the chemistry of these fossils. And that allows us to do things like look at the rise and fall of different evolutionary lineages through time. So over on the left here, my own personal favorite groups of fossils, the trilobites, which reach a peak in diversity about a little more than 400 million years ago, and exist for quite a long period of time before they finally become extinct about 250 million years ago. So these are relatives of modern crabs and spiders and other arthropods. And then in the middle, the ammonites, these are relatives of squid that actually coexisted with trilobites, but then existed long enough to eventually also coexist with dinosaurs much later, before finally disappearing from the record about 66 million years ago. And then on the right here, this is something that some of you may recognize from walking California beaches. This is a sand dollar, which is a member of a broader group called echinoids that, as far as we can tell, are as diverse now as they have ever been. So we can ask questions about what are the processes that drive the rise and fall of these different lineages through time. The other thing that this figure really nicely illustrates is one of the most striking patterns in this record, which is that diversity tends to rise gradually through time and then fall very rapidly. These are the major mass extinction events. And the modern era of study of mass extinction events really came from work done here in the 1980s and 90s at Berkeley by Walter and Louis Alvarez, focused on the most recent major mass extinction, the Cretaceous Paleogene here, the extinction in which the ammonites and the non-avian dinosaurs and a lot of other groups became extinct. And they famously showed that this was caused by a very large extraterrestrial impact. The largest of all major mass extinctions, the Permian-Triassic so-called Great Dying, in which the trilobites finally disappeared, we now know was caused by a massive injection of greenhouse gases from the largest volcanic eruptions of the last 500 million years or so, with consequent extreme warming and ocean deoxygenation, acidification, and many other things. Then the third major extinction here, the late Ordovician mass extinction, is kind of an oddball, and one that we still don't understand quite as well as the others. It coincides with this very unusual and abrupt cooling and glaciation event, but there are still many open questions about what caused it. So just in the last couple of minutes, I'm just going to walk you through how we approach trying to understand these events with a focus on the late Ordovician mass extinction. So a lot of what we do is just based on sort of good old-fashioned paleontological fieldwork. We go to places where there are a lot of fossils. It's a place called Anticosti Island that preserves a very rich record through a period of time before, during, and after this mass extinction event. And we work our way up through the rocks, collecting lots and lots of fossils, and that allows us, on the left here, to make a figure like this that delineates when different species first appear in the record, the kinds of habitats that we find them in, and when they disappear from the record. And when we do this, for example, we can see that a lot of these species are disappearing from the record within or just below this bed here that I've highlighted in red. Then at the same time, from those same collections and often from the same fossils, we measure all kinds of different aspects of chemistry, stable isotope ratios, trace metal ratios, that can inform our understanding of environment through time. The figure on the right here is just mapping out different elemental concentrations in a fossil coral here. This is something we do to sort of screen for preservation and pick the best preserved fossils. And then based on that, we can delineate trends through time. And I'm not going to go into details here, but one of the things you'll notice is at that same level where a lot of species disappear, we have big changes in a number of these different geochemical proxies. And in particular, this implicates a big perturbation of the global carbon cycle and a quite substantial cooling of the tropical oceans. So we can put these together and again, note that they correlate quite tightly, but if correlation famously does not equal causation, causation is a trickier issue. And the way that we typically try and get at this is through making predictions of selective extinction survival. So every possible scenario for what might have caused an extinction makes some prediction about the kinds of species that ought to have done well, the kinds that should not have done as well. And so we make predictions, we do statistical analyses, and then we compare those to models. So this is it's a figure from a paper we're about to send in, where we look at the distribution of this group, the planktonic graptolites, which were very widespread in the late Ordovician oceans, and then suffered very high rates of extinction. We simulate a whole variety of different possible types of perturbation, and then we see which ones best match the record. And in this case, we can see that this temperature drop that we estimate from the Anacostia Island record predicts actually quite severe extinction of this particular group. So may actually be one of the drivers that's implicated in this event. And so with that, I'll just show a couple of pictures of some of the fantastic students who have been involved in this work out in the field, and would be happy to take any questions.
Thank you so much, Seth. So we have a few minutes for questions for audience members. I request that you add your questions to the chat. And I'll just get started with one of my questions. So, I mean, one of the key things for your work is that you have to find fossils, right? And so when you stop finding fossils of some species, what does that mean? Does that mean that that particular species just went extinct? Or are there other things you need to worry about having an influence?
Yeah, no, that's a great question. And so again, what we actually observe is the absence of fossils. And what we would want to actually know is when did that lineage really went extinct? But of course, we're always, we're only a small number of individuals, even in groups like clams and corals that have a high probability individually of ending up as fossils. We still only get a small number of them. And so one of the things we can do is look at their distribution in the record before they go extinct, and look at basically how gappy is it? Are they kind of continuously present, or are there long gaps? And that allows us actually to put confidence intervals on how comfortable we are saying this particular species is now extinct because we're no longer recovering it.
Right, got it. Okay, and so now we'll move on to a couple of questions from the audience. And the first one is from Marshall Byrne. And the question is there a way to test the snowball Earth hypothesis with fossils?
So a little bit, unfortunately. So snowball Earth events, for those who don't know, are these really profound, probably the most profound climate events in Earth's history, where, as far as we can tell, almost essentially the whole planet froze over for, in some cases, tens of millions of years. It would be great to be able to do this. The problem is, one of the things that makes the snowball events interesting is that they precede uncontroversial evidence of animals in the fossil record, and certainly of skeletonized animals of the kind that leave these sort of chemical records for us to study. So unfortunately, one of the reasons that snowballs still remain very mysterious is a lot of the tools that we bring to bear on understanding climate over the last 500 million years are not available to us when we don't have skeletonized animals and algae in the oceans.
Okay.
It would be great, though.
Yeah. So moving on from the snowball Earth to another question about extinction events. So this one's from Alistair Fraser in the audience. And so their question is, are there any mass extinction events that are not correlated with temperature change? So everything we've discussed so far, there's kind of drastic changes that have happened. Is there something else that does not correlate with massive
That's a really good
fluctuations?
question. I mean, so in some ways, the Cretaceous Paleogene would be the example. It's a little bit, it's interesting because it was sort of the first one for which we had a good sense of the causality. And for initially, a lot of people said, oh, maybe they're all big asteroid impacts, but that hasn't really held up. But there is, you can't see the temperature change on the scale of that figure that I showed. But we do have some evidence and pretty strong support from models saying that probably one of the big agents of extinction after that event was a so-called nuclear winter, where you loft so much material up into the upper atmosphere that you block out the sun for a substantial period of time. So there almost certainly was quite extreme but very short-lived cooling, and maybe more important than that, productivity collapse from blocking out the sun. But that's sort of a secondary consequence of the impact.
Yeah. Okay. And then one final question before we move on. So we've been talking about events in the past, and you've been mentioning that the biggest extinction event was caused by a massive injection of greenhouse gases into the atmosphere, and we know that that's along the lines of what's happening now. But how do those two compare, and how worried should we be? This also kind of touches upon a question from the audience member saying, "What do your studies say, if anything, about the likelihood of human extinction?"
All right. Well, I'll take the first one first. So it's very striking and a little bit worrying that this big extinction event, which occurred long before humans were on the planet to do any of the things we're doing, seems to have the same sort of fundamental cause as the current climate crisis, which is rapid injection of greenhouse gases. It's very hard to compare rates. Sort of measuring rates in the deep geological record is a notoriously difficult question. So, but to the extent that we can, the suggestion is that actually current rates are probably higher, if anything. So that's worrying. But on the other hand, there are a lot of ways in which the Earth system has changed since the Permian mass extinction that I think probably make it a little bit harder to perturb as extremely. So there have been major changes to the carbon cycle, for example. There's a big deep marine carbon reservoir that didn't really exist at that time. The Earth is probably better oxygenated in general, or the oceans are better oxygenated. So I'm sort of optimistic that I don't think we can reproduce the Permian Triassic mass extinction. But the Permian Triassic mass extinction is the worst thing that's happened in the last 500 million years. So that's maybe cold comfort to some degree. And then, I mean, human extinction, on a long enough time scale, everything ends badly. But I don't, I don't know that I have a useful opinion on that, except to say that Earth history tells us that when we do the things that we're doing now, we have reason to think there are going to be some pretty severe consequences.
Oh, thank you again, Seth. This was truly fascinating. So we will have some more time at the end to address more audience questions, so please keep them coming. But for now, let's turn to our final speaker, Associate Professor of Earth and Planetary Science, Daniel Stolper. Daniel is a geochemist with interests in a variety of problems related to the rock record, to microbiology. We collaborate on microbiological questions and biogeochemical cycles, both in the present and in the past. His approach to scientific problems is centered around linking experiments, physically based models, and natural observations in an integrated fashion. And if necessary, as you'll see today, he develops new methods and techniques. In addition to being a professor at UC Berkeley, Daniel's also a faculty scientist in the Earth and Environmental Sciences area at Lawrence Berkeley National Lab, which is right up the hill from our UC Berkeley campus. So Daniel, can you tell us more about your research?
Of course. Hello, everyone. Today I'm going to talk a little bit about my research at the end, which is trying to take here a bristlecone pine and use it as a way to reconstruct the humidity of past environments. But in doing that, I also hope to teach everyone a little bit about isotopes. You've heard a little bit about this, and why they're useful in general for understanding Earth history, life, things like that. So I want to do first to just take a step back and talk about isotope geochemistry. I call myself an isotope geochemist, and just make sure we're all on the same page of what is an isotope. And I want to start with an example of oxygen. So we're all breathing molecular oxygen right now, O2, I hope. And there are three different flavors of this that we see commonly. When we talk about an element, the element of oxygen, what we're talking about is the number of protons in the nucleus, and here are the electrons. But that nucleus, except for hydrogen, has another particle in it, neutrons. And the number of neutrons sets up the isotopic composition, these different flavors. Oxygen 16, which is most of the oxygen in our atmosphere, oxygen 17, which is very rare, and oxygen 18, which is rare. They're all oxygen. They all behave in similar ways, but they have ever so slightly different chemistries that we can probe and understand and measure. And just to connect this back to something that Jenny talked about early on in terms of cosmic chemistry, a long-standing area of research is measuring ratios of 018, 16, and 17, and 16 of our solar system. Here's the sun, all these different meteorites. Here, Earth is right here, and the ices that we think were in the early solar system plot way off scale, and trying to understand the mixing of these different components as we generated the big terrestrial bodies, the meteorites, and the waters. So one of the things that most people in my experience are familiar with when talking about isotopes are things called unstable or radioactive isotopes. Any of you who saw what I thought was a wonderful movie, Oppenheimer, all about UC Berkeley, in my opinion, this was all about the isotope uranium-235 and the goal to enrich uranium-235 to make a bomb. Another isotope that you'll hear about a lot is radiocarbon or carbon dating. So here are the Dead Sea Scrolls. You can measure this radioactive carbon isotope, C14, and actually get the ages of ancient carbon-bearing materials. And Berkeley has a very long history in fundamental observations and measurements and technical developments in the history of isotopes. So one is William Gioch, who's a Nobel Prize winner in chemistry, discovered oxygen 17 and oxygen 18 in the late 20s. At Berkeley, C14 was created. And this was used by Calvin of the Calvin cycle to discover the Calvin cycle. So this is how he actually figured it out. Berkeley was deeply involved in that story of the enrichment of uranium beyond things going on in New Mexico. Lawrence built these giant instruments called calutrons after Cal, in how we actually separated out U-235 and U-238. And we also used it to date here at Berkeley, fundamental evolutionary histories of hominids. This is a date early on of early human ancestors that changed our evolutionary timeline by about 1 million years. Right? So what do? I don't do that stuff. I don't build any bombs or anything like that. What I work on are things called stable isotopes. These are things that do not decay, as far as we know. Once made, they sit around. And what we use them for is to learn about chemical and physical processes going back in time as well as today. So I just want to give you an example of something you can do with oxygen isotopes. This is 018 to 16 ratios. Don't worry about the numbers, just look at the colors. This is in water, rainwater across North America. The isotopic composition of rainwater, its 018 to 16 ratio, varies as you go from equatorial regions up to Greenland and Upper Canada. And this happens for a variety of things that we might want to learn about. The source, mountain range and elevation modify this. The amount of rain modifies this. And as we'll come to later on, the amount of local evaporation modifies these isotopic compositions. So why? I just want to give you a taste of the physical and chemical basis for this. When we make rainwater that falls on land, it all ultimately comes from the oceans. And it turns out that when you evaporate water from the oceans, oxygen 16 is easier to evaporate than oxygen 18. And it makes our clouds different in their isotopic compositions. When that cloud moves onto the continents, O18 prefers to come out in the water. And so it falls and changes its isotopic composition as you go along. And these cartoons are all actually related to very fundamental chemical behaviors related to the energy of oxygen 18 versus oxygen 16 that change how they move from a reactant to a product and go through a transition state. And this is the tools of quantum chemistry, statistical mechanics that we use to really get at the physical fundamental processes going on. But don't worry, we're not going to go into the quantum mechanics of this in any detail. So I just want to give you a few examples of things you can do with isotopes, stable isotopes. So one is reconstruct ancient temperatures. And Seth alluded to this a little bit. One example of this is not my own research, is people have taken snow from across Antarctica, going from the edge where it's warmer to the deep interior, to the South Pole where it's very, very cold. And there is an observation that we understand, but it's just a physical observation, that the temperature of Antarctica, where the snow is collected, has a clear linear correlation with the 018 to 16 ratio of the snow. And regardless of why, you now have a thermometer in here. You can measure the isotopic composition of the snow and go back and get temperature. And this is one of the most important records of temperature over the past million years based on ice cores. So there's been huge programs by us, the Europeans, the Australians, people in Asia and China, of doing deep drilling cores of ice, extracting them and understanding them. You get both the ice, but you get preserved in these ice cores bubbles of ancient air. And so you can link our atmospheric composition in the past to compositions, structures of the ice. An observation is if you take those isotopic records, take that line and calculate a temperature, you can measure from today to 400,000 years ago. In this example, change in temperature in Antarctica. So here's today, last ice age, it was cold by 8 degrees. It was warmer going backwards in time. And if you take these and compare it to the amount of CO2 in these records, they directly correlate. When it's warmer, there's more CO2. When it's cooler, there's less CO2. And so from the isotopic composition of the ice, we can directly link what we talk about with climate, we talk about with global warming, to a past record where we've seen these sorts of variations. And this links back to something Seth talked about when Seth was going through our understanding of climate history and evolution of animals and diversity. We have records of temperature over the past 500 million years, the global mean surface temperature, that 500 million years ago, it was 32 degrees apparently, compared to around 15 today. And these changes link up to, in some cases, mass extinction events. Most of these climate records are based on isotopic compositions and our reconstructions of them. What else can we do? This is a fun thing. You can do forensics with this. So the isotopic composition of the water you drink goes into your hair. And so this is a map of the isotopic composition of people's hair depending on where they live. And what's interesting about our hair is it also is a time recorder. The hair at the base of your root is newer, more recent than at the tip. That is very old hair, 6 months old. And there's complexity with this of how your hair grows and things like that. But you can actually learn about where someone has been based on their hair. This is an example of someone who was living someplace with an isotopic composition of around 12. That would be here in Berkeley. And then at one point they went and got an internship. I'm making this up, an internship, and they started having drinking water locally. The internship's probably over here someplace, and their isotopic composition of their hair changed. right? And you can see this in the record. And this is actually something the FBI does. They have instruments to measure the isotopic composition of hair. And that's because sometimes there is what's called a Jane Doe, a body where they don't know who it is. And they want to know, was that person living there or were they moving around? So you can take hair and you can say this person was actually moving around based on their isotopic composition and try and figure out where they may have been prior to the murder. And so there's a whole bunch of fun things you can do with this. And so now I want to take you through an example of using isotopes that's going on in my lab right now. And I want to go after the question and the age-old adage of it's not the heat, it's the humidity. The people on the East Coast are going to start boiling up. The trash is going to smell in New York City. Why is that happening there versus the nice dry temperatures over here? And so when we talk about climate, often we talk about temperatures. This is a measure in Celsius of temperature. Here's us, our body temperature is around 37. We often worry about T as a driving force of change. And we even saw that, I'm not being critical of him, but saw that in Seth's talk as a fundamental variable. And it is. But on land, that also is coupled to humidity. There is a set of studies going on right now, and this is known for a while, but we will survive better if it's hot and dry because we can sweat and get rid of heat compared to if it's hot and humid. So global warming in a place that's humid is going to bring us potentially into a critical zone where we can't dissipate our heat and we're all going to be in trouble versus if you're in a dry place, you can survive to a higher temperature. And this has actually been projected in climate models of annual hot hours here going up where people will not be able to work during the day. And so India has huge issues of projected climate change of 2 to 4 degrees. We have issues in the eastern part of the United States. In the Gulf, we have these places. In East Asia, we have them. In Africa. And so this is a very serious concern about future climate, not just in temperature, but in humidity. So how well do we know this about the past? I'm always very interested in thinking about, just like what Seth talked about, which I talked about, how are things happening not just in the modern, but in our history and understanding of the evolution. These are maps of what climate was like during the last glaciation 20,000 years ago. There's snow in Canada coming down into the United States. We have records of climate and humidity, but they're very coarse. They're things like in blue, wetter, or in red, drier. It's not quantitative, 20% humidity, relative humidity, 30%. And so what I want to do today is establish with everyone here something going on in my lab where we want to take our trees and try and convert them into what you can buy at Home Depot for $10, which is a hygrometer, something to measure the humidity in a given environment. And the way we're going to do this is using wood and using trees which have evolved to respond to change in humidity, change in the environment, and now we're going to use them to reconstruct it. And so when you think about what is the job of a tree, the job of a tree is to do photosynthesis. So one thing they want to do is bring in CO2 from the atmosphere into their leaves. But in doing so, they also need to move water through their system. And so there's always a tug of war in water going through the plant, being lost and them drying themselves out versus getting in CO2 to do photosynthesis. And we know if you look at leaves around the world that very dry places have very high 1816 ratios. The evaporation controls the isotopic composition in the leaf. So places in the west of the United States, the middle of Australia, here in the Gulf, leaves are very high in 1816, whereas in warm but wet places like in the Amazon, you don't get that enrichment. So built into the isotopic composition of leaves is telling you something about the humidity. And that water that's in there is incorporated into wood that is preserved for, in many cases, millions of years. The oxygen of cellulose, our paper, is made from oxygen that's created in the leaf. And so one of the things that I have done in my lab is try and develop ways to measure the isotopic composition of ancient cellulose and build up a hygrometer, a way to take old wood and learn about ancient humidities. And this is very much a lab-based experiment. We built and designed a brand new instrument, a whole automated robotic system to do instrumentation, and then we measured the isotopic compositions effectively in the same way Jenny talked about it for looking at things in space. There are spectral features when we shine light on CO2 in which we can measure O18, O17, and O16 very precisely and very, very quickly. All right, and so I just want to show you, does it work? All right, does this thing actually work? We have used a great collection of wood housed in UC Berkeley's collections, where in the 30s we went around, we, UC Berkeley faculty, collecting wood to archive them and understand them for the lumber industry. This is a set of records of a compilation of that data in which we're looking at relative humidity known and the isotopic composition of O18 only. And indeed, there's a very nice relationship in which we could take the O18 and learn about humidity. We've also gone a step further and use all three isotopes of oxygen, which has never been done, O16, 17, 18, which is actually a better way, but harder, in which we can measure these compositions and again, reconstruct humidity. And so we believe that we have a real paleohygrometer based on wood. So what are we doing next? Now is the fun part of applying this to systems. So one is we're using this great collection of wood housed in the UC Berkeley archive to expand this. We're also using University of California reserves where we can go and core wood, study them, and understand what's going on in the system. We have ancient samples that we've collected. We're working on samples as an example from the La Brea tar pits. And just note, this was originally studied by UC Berkeley. And then we're using greenhouse experiments to really narrow our understanding of what's going on here at UC Berkeley. With that, I hope I've convinced you that isotopes are interesting, can teach you something, that humidity is equally important as temperature when we consider climate change. And with that, I'm happy to take any questions.
Thank you, Daniel. All of these presentations have been fascinating. And so with the time we have left, what I'd like to do is invite all of our speakers back for some last questions. I'd love to take any questions from the audience. And just to get going, I guess one of the questions that I have for all of you is, what do you think are some of the exciting new directions for geobiology at UC Berkeley? So maybe we'll start with Jenny. All right, let's start with Daniel, actually, since you're the last speaker.
So I would say there's a lot of exciting directions, but one of the things that I would say that's come out that's very much on the minds of people here at Berkeley is the advances in genetic editing. Genome editing is something, Dipti, you could answer this question, but that we can now manipulate proteins, we can manipulate the expression of them, we can build ancient proteins and resurrect them. We can do Jurassic Park. And so there's a real, to me, future of using the modern tools that are emergent of molecular biology that are changing health sciences and apply them to the study of life's history in a really different and special way that Berkeley, through the advances of Jennifer Doudna, Jill Banfield, things like that, will let us be different in.
And better than
Jenny?
Jurassic Park, hopefully.
Yeah.
Jenny, do you have anything you're particularly excited
I'm
about?
always excited about research going on at interfaces between fields. And I think, as we've seen with the 3 talks tonight, there's a lot of variety in the kind of types of questions people are asking related to geobiology, but getting everyone like this together in the same room to think about what kind of new interfacial questions we can ask, I always think is really exciting.
And Seth?
I mean,
Any
yeah, I
thoughts?
will second all of that. I happen to be particularly excited at the moment about some ongoing projects involving Daniel with some emerging isotopic techniques that are going to help us reconstruct sort of not just species, but ecosystems and trophic interactions in deep time, which is really exciting because, when we talk about these mass extinction events, sort of talk about them like everything is just forced by the environment, but ecosystems are very complex. So a lot of nonlinear dynamics that come into play that we can't understand unless we have that kind of information. And then there's also a line of evidence that we haven't even talked about today, but that also began at Berkeley and is starting to come home to Berkeley, which is we're starting to build an ancient DNA facility in this building that I think is also going to open up a lot of really exciting possibilities.
That's amazing. I'm afraid this is all the time we have. Once again, thank you to our speakers for their enlightening presentations. To close, I'm going to hand the floor back over to Dean Kahn.
Thank you, Dipti, for guiding such a wonderful conversation tonight. And my deepest thanks to our speakers, Jenny, Seth, and Daniel, for sharing your extraordinary research with us. When we began this evening, I mentioned that geobiology allows us to look at the coevolution of Earth and life. Tonight, we've seen exactly how vast and interconnected that story truly is. This incredible breadth of research is a perfect example of why basic science is so essential. Thank you all for spending your evening with us, for your fantastic questions, and for your continued dedication to basic science. Your engagement and support empower our faculty and students to keep illuminating the unknown. We look forward to seeing you again for the next Basic Science Lights the Way, which will be on Wednesday, May 6th at 5 p.m. 4 Berkeley astronomers, one of which is me, will share how they are using the Vera Rubin Observatory in Chile, which contains the largest camera ever built, to bring cosmic mysteries to light. Please reach out if you would like to learn more about anything we've covered. As always, a video of today's session will be available on our website, basicscience.berkeley.edu, approximately 2 weeks from now. And you can always visit the website to watch episodes missed. Until next time, stay curious, fiat lux, and go Bears.